Hello, my name is Joshua Mugabi. I welcome you to Karyotype and Genetic Diseases. What is karyotype? The name karyotype is given to the set of chromosomes of an individual, usually when visualized and identified under the microscope. The visualization generally is made with the cells in the initial phase of cell division, for the chromosomes to be seen already replicated and condensed. Which type of genetic disease can be identified from the visual analysis of the number of chromosomes present in a karyotype? The counting and identification of chromosomes in the karyotype of an individual can diagnose the aneuploidy diseases caused by alteration of the number of chromosomes in relation to the normal number of the species. Why, in preparation of a karyotype analysis, is the use of the substance like colchicine interesting? Colchicine is a substance that disallows the formation of microtubules and thus of spindle fibers in cell division. Under action of this drug, the cell interrupts division at metaphase, and the anaphase does not occur. Therefore, use of colchicine in the study of karyotype is interesting because chromosomes will be seen replicated and condensed. What is the karyotype found in Down syndrome? Down syndrome is aneuploidy, that is, numeric alteration of the chromosomes within the cells compared to the normal number of chromosomes of the species. Affected individuals have their cells an additional chromosome 21 instead of only one pair. For this reason, the condition is also called trisomy 21. The affected person has karyotype with 47 chromosomes, that is 45 plus XY or 45 plus XX. What is aneuploidy? What are the conditions caused by aneuploidies? Aneuploid is an abnormal number of chromosomes in the cell of an individual. The main aneuploidies of the human species and their respective conditions are nullisome, absence of any chromosome pair of the species, often incompatible with life. The monosome, absence of a chromosome from a pair. For example, Turner's syndrome, that is 44 plus X. Trisomies, that is an extra chromosome. For example, triple X syndrome, 44 plus XXX. Or Edwards syndrome, trisomy chromosome 18, that is 45 plus XY, or 45 plus XX. In general, what is the cause of aneuploid? Generally, aneuploids are caused by impaired assortment of chromosomes during meiosis. For example, when homologous chromosomes of the pair of 21 do not separate, gametes with 2 chromosomes 21 and gametes with chromosome without chromosome of 21 form. If a gamete with 2 chromosomes 21 fecundates a normal gamete of the opposite sex, the zygote will present trisomy. 3 chromosomes 21. If the gamete without chromosomes 21 fecundates the normal gamete of the opposite sex, there will be a zygote with monosomy, with only 1 chromosome 21. The defects in the separation of chromosomes during cell division are called chromosome non-disjunctions. During meiosis, non-disjunctions may occur in the anaphase 1, non-disjunction of homologous chromosome, as well as in anaphase 2, which is a non-disjunction of the sister chromatids. Do all genetic diseases result from alteration in the number of chromosomes of the cells? Besides aneuploids, there are other genetic diseases, other chromosomal abnormalities, and also genetic mutations. How are genetic diseases classified? Genetic diseases classify into chromosomal abnormalities and genetic mutations. Among chromosome abnormalities, there are the aneuploidies diseases caused by alteration of the normal number of chromosomes of the species euploid. For example, aneuploid in Down syndrome, or what we call trisomy 21, in which there are 3 chromosomes 21 instead of the normal pair. In the group of chromosome abnormalities, there are also deletions, absence of part of a chromosome. The inversions, in which the chromosome breaks and its pieces reconnect in the inverse manner. And the translocations, pieces of the chromosome that exchange positions. In the genetic mutation group, there are the deletions, 1 or more DNA nucleotide absent, the substitutions, and the insertions. What are genetic mutations? Genetic mutations are alteration of the genetic material compared to the normal condition of the species. And this involves modification of the normal nucleotide sequence of a gene, but without structure or numerical chromosome changes. These modifications may be deletions, loss of a nucleotides, substitution, that is exchange of nucleotides by other different nucleotides, or insertions, placement of additional nucleotide in the DNA molecules. Does every gene mutation cause alteration in the protein the gene normally codifies? Not every gene mutation causes alteration in the composition of the protein the gene codifies. Since genetic code is degenerated, that is, there are amino acids codified by more than one different DNA nucleotide triplets. If by chance mutation substitutes one or more nucleotides of the codifier triplets, the newly formed triplet still codifies the same amino acid codified by the original triplet. There will be no modification in the protein made from the gene. How do genetic mutation influence biological diversity? Too extensive, too frequent genetic mutation generally are deleterious for individual and species. These mutations often cause important phenotypic changes or defects incompatible with the survival of the body and the continuity of the species. However, Small genetic mutations that do not cause the appearing of lethal changes are continuously accumulated in the genetic patrimony of the species. These mutations gradually add to each other, giving birth to small phenotypical changes in individuals. These small changes are exposed to the selective criticism of the environment, that is the natural selection, and the more favorable for survival and reproduction are preserved. The remainder are eliminated as their carriers have difficulty in surviving and reproducing. In this manner, the combined processes of accumulation of the small mutations of unnatural selection incorporate new features in the species, and they may even lead to speciation, formation of new species, and promotion of biological diversity. Obviously, only genetic mutations transmitted by cells that originate new individuals in sexual and asexual reproduction have evolutionary effects. What are mutagenic agents? Mutagenic agents or mutagens are physical, chemical, or biological factors that can cause alternation in the DNA molecule. Examples of well-known believed to be mutagenic agents are X-rays, alpha rays, beta rays, and gamma rays, ultraviolet radiation, nitrous acid, dyes, some sweeteners, some herbicides, many substances of tobacco, some viruses like HPV, etc. Small DNA fragments known as transposons can also act as mutagens, and when incorporated into other DNA molecules. How are mutagenic agents related to cancer incidence in a population? Is cancer a disease transmitted to the individual offspring? The exposition of a population to a mutagenic agent, for example, people living in surrounds of Chernobyl nuclear power plant and exposed to the radiation from the nuclear incident of 1986. This increases the cancer incidence in that population. This occurs because mutagenic agents increase the rate of mutation and the probability of a mutant cell to proliferate in pathological manner. Cancer. Cancer itself is not hereditary, transmissible disease. Genetic predisposition for the development of cancer, however, can be inherited. How do the repairing enzyme of the genetic system act? There are enzymes within the cell that detect errors and alteration in the DNA molecule and begin a repair of these errors. The first enzyme is known as restriction endonucleases, specialized in cutting DNA molecules, also used in genetic engineering. Cut the affected piece of DNA. Then polymerase enzyme builds correct sequence of nucleotides correspondent to the affected piece, taking as template the DNA chain complementary to the affected chain. Finally, a new correct sequence is bound in the DNA under repair by specific enzymes. What are some diseases or genetic abnormalities caused by recessive genes? Examples of recessive genetic diseases, we have cystic fibrosis, albinism, phenylketonuria, galactosemia, Tay-Sachs diseases. What are some diseases or genetic abnormality caused by dominant genes? Why are severe dominant genetic diseases rare than recessive ones? Examples of dominant genetic diseases are Huntington's disease, Huntington chorea, neurofibromatosis, hypercholesterolemia, polycystic kidney disease. Severe and early autosomal dominant disease are rare than recessive autosomal disease because in this last group, the affected allele may be hidden in the heterozygous individual and transmitted to the offspring until undergoing homozygosity, actual manifestation of the disease. In severe dominant disease, the heterozygous manifests the condition and often dies without having offspring. Some genetic diseases are of later manifestation, like Huntington disease. In these cases, the incidence is higher because many individuals have children before knowing that they are carriers of the dominant gene. What is consanguineal marriage? Why is the appearing of genetic diseases more probable in the offspring of consanguineal marriage? Consanguineal marriage is the marriage between relatives, that is, people having a common near ancestors. The consanguineal marriages increases the probability of recessive genetic disease in the offspring, since it is common for the people of the same genetic lineage to be heterozygous carrier of the allele. That condition, recessive genetic disease. How the early diagnosis of genetic diseases usually done? Genetic disease may be diagnosed in prenatal period by karyotype analysis in the case of aneuploid, or by DNA analysis in the case of other diseases. The test is performed by removal of material containing cells of the embryo by amniocentesis, extraction of amniotic fluid, or chordocentesis, puncture of the umbilical cord, or even by chorionic villus biopsy that can be done earlier in gestation. Ultrasonography is the diagnostic procedure for some genetic diseases that produce morphological variation during embryonic development. The study of genetic family trees is an important auxiliary method in the early diagnosis of many genetic diseases. Thank you very much for participating in karyotypes and genetic diseases. Please do like, share, and subscribe.